Now let's study about the green fluorescent protein. If you go to the aquarium, usually we see this kind of a beautiful jellyfish. And some of those jellyfishes, they emit light with different colors. One Japanese scientist got interested in this phenomena and he tried to isolate out this protein. He spent a good amount of time and then isolated out this protein, so-called aquarin, and this is the structure of the protein. After he purified this protein, he found something strange. Originally, the jellyfish shows this kind of a green-blue light, but if you irradiate with a UV light, it generates this kind of green color. But this aquarin, it generates only blue colors. And the mechanism of this aquarin is bioluminescence. That means utilizing ATP and it generates blue colors. Then where is this green color come from? He studied further and he found another protein, so-called green fluorescent protein. Now this is similar size to aquarin, a little bigger, and very interestingly, this protein is not bioluminescent protein. This protein is fluorescent protein. Fluorescent means the input is not bioenergy like ATP. Input is light and output is also light. That's the fluorescence. Now inside this jellyfish, aquarin generates blue color. And then that blue light can be transferred to GFP. GFP accepts that blue color and then lose a small part of that energy and then emit green color. And that's why this GFP looks like green. Seems like a jellyfish has both of these proteins and sometimes they look a little more bluish and sometimes they look more greenish. And among these two proteins, which protein is more interesting to you? In other words, if you utilize this protein for other study, which can be more useful or more convenient to utilize? In aquarin case, you may need to add ATP to turn it on. In GFP case, you can activate that protein by giving light, which may have more useful applications. Can you guess? The Nobel Prize was given to GFP, and these three people shared that Nobel Prize in 2008. And the middle person here, Dr. Osamu Shimomura, he is the person who isolated this GFP and cloned. Then how about the other two people? The first one here, Martin Charpy. He got an idea that maybe this protein can be transferred to other life form and then can express that fluorescent property in other life. He realized that crazy idea in C. elegans, and he put that GFP gene in C. elegans and demonstrated that, yes, those fluorescence can be expressed in different life form. Image quality is quite bad, but this historic paper has been published in Science, and this was his main contribution for the Nobel Prize. How about the third person? We had seen this guy before. Dr. Roger Chen, he was the scientist who developed the calcium dyes. He analyzed this GFP with a chemist view. The center part of the GFP, this chromophore is composed by 3 amino acids conjugated. And by looking at this structure, he got an idea by changing those environment of this protein, maybe he can change the colors of this protein. So originally the protein was a GFP. Now he made a broad range of different colorful fluorescence proteins and gave a name as BFP, blue, cyano, green, and yellow, and red color. There are too many red colors. He gave a tomato, cherry, raspberry, plum. And his chemical intuition how to make this kind of colors awarded him the Nobel Prize. If you express different GFP derivatives with different colors for different bacteria, now you can draw this kind of colorful pictures with the bacteria. Or you can transfect those genes in fish. Now we have this kind of a glow fish, not glow stick. Even that GFP protein can be expressed in monkey. Monkey is maybe closest cousin to human being, and monkey is possible. Technically, why not human being? So it seems like there is no limit of application of this GFP protein across all the species. Now, coming back to the original topic, if you want to visualize only one protein's localization. Previously we have seen utilizing antibody, and the problem of antibody was you need to kill the body. How about utilizing GFP? Let's say this is your target protein. Then you add GFP to your protein. Then maybe your protein carries this fluorescent protein. It can visualize its site. Now, genetically, how can you engineer that? Let's say this is your target protein, and this is the gene. This is a protein part, and this is stop codon. And here, you can add GFP's genetic code. Then, the protein can be synthesized, and at the end of the protein, GFP can be added. Utilizing this powerful genetic engineering technique, now it is very possible to stain specific proteins in specific area with different colors. Now you can see this kind of a beautiful multicolor imaging in C. elegans. It's a lot more improved than original C. elegans images, isn't it? Similar things can be applied to mammals. Now you can express that GFP protein in specific locations in the animal is also possible. This GFP technique has been applied to cell cycles. Cell cycle has different stages. In the beginning, the cells are preparing for cell division, and later it enters into DNA synthesis and real mitosis. For each stage, there is a specific checkpoint proteins, and G1 can be modified with LFP, and G2 can be modified with GFP. Then it is possible to see those cell stages with different colors. Then it's very easy to see G1 is red color, and then from S, G2, M stage, this is green color, and in the middle, this can be yellow color. This is just a snapshot, but if you see those dynamic movie of this cell cycle, it's amazing. So let me show you one movie clip to show this dynamic cell cycle. It's quite beautiful, isn't it? And this is the beauty of to see live cells in dynamic status. One more example I want to show you for very complicated structure of neurons. Brain has so many neuron cells. That's why to figure out those connections is quite difficult because of too many numbers. In that case, labeling of each different neurons with different colors can be really helpful to analyze those connectivities. Here is another beautiful trick, so-called the rainbow. Let me introduce this to you with another beautiful movie clips.
Although we already know a lot about our brains and brain cells, we're still figuring out where different individual neurons are positioned in the brain and who they connect with. It's like trying to figure out how a big city operates at a distance. Even if we zoom in pretty far, it's still hard to make sense of what we're seeing without better tools. To better understand our city, having a map of the roads and streets would help to identify important city structures and figure out how different parts of the city are connected. Based on the connections, we can then make educated guesses about the kinds of human activity that might happen in different areas. Just like Google Maps for a city, neuroscientists have been working on a connectivity map for the brain. This connectome will help identify how neurons physically connect to one another. This is no small task. If we take a satellite view of the Earth, it's hard to see where roads lead without visual labels. Similarly, it's hard to tell one neuron with its many twisting branches apart from its neighbors and their branches, because neurons are all the same color. To shed some light on the situation, neuroscientists have come up with a way to brighten the brain with an artistic flair in mice. They do this by allowing individual neurons to make their own special mix of fluorescent protein paint, producing a rainbow-colored brain. This brainbow technique lets us separate individual neurons by color. Think of it as color-coding neurons so we can easily see where they are, just like color-coding transit lines on Google Maps. A brainbow is made possible by what's called Cre-Lox recombination. Here's a quick and dirty explanation of how the latest and greatest brainbow is made. We give neurons the genetic equivalent of a paint pot set. Each pot contains the DNA recipe for a different color of fluorescent protein. This paint pot set has cutting sites between each pot of paint, called LOX-P sites. A protein called Cre will randomly choose a pair of LOX-P sites to cut, and will remove whatever paint pots are to the left of the cut. The neuron will only read the DNA recipe of the very next paint pot in what remains of the set. So, through the action of Cre, each paint set will produce only one randomly determined fluorescent protein color. When we give each neuron many paint pot sets that each contribute a randomly determined color, every neuron will end up creating its own unique mixed color from the random ratios of their original fluorescent protein paints. These neurons are like tiny painters, mixing basic paint colors to create a new complex shade. Now we can tell almost 100 uniquely colored neurons apart in these beautiful brainbows. Brainbows are not just nice to look at. They differentiate neurons from their neighbors and allow scientists to follow an individual neuron's branches, the neurons' axons and dendrites, as they crisscross over other neurons' branches and reach their final destinations and connection partners throughout the brain. In the end, knowing where the subway lines in a city go won't tell us how often the trains are running, or why they are on a particular schedule, or if the subway cars might break down or change direction. Likewise, knowing where a neuron is located and who it connects with will not tell us everything there is to know about that cell's function and role in producing thoughts, memories, or disease. However, the brainbow method of labeling neurons is a valuable tool that makes it possible to create detailed maps of neural connections and provides a powerful and beautiful foundation with which to understand the basic structure of the brain.